So good afternoon, everybody, and welcome now to our second class in biology, general biology. Today we're going to move on to start discussing some of the chemistry of life, biochemistry. We only do it in really simple terms. Biochemistry is a huge science. You could study for years, but we do brief introductory set of lectures to introduce you to molecules which are important to life. And the principal molecule which is important to life, of course, is water. You've got to get used to the way I say it. I can't say it the American way. But water is essential for life because all metabolic processes in the cell take place in solution. So it is essential for life that there be liquid water available. Now, if we look at other planets, look at Venus, which is closer to the sun than us. There's plenty of water on Venus, but it's all in the form of water vapor. It's a gas. It's not available to make solutions. Look at Mars, which is further away from the sun than us. By and large, Mars is either too hot or too cold for liquid water to occur. There is liquid water, there is solid water in the form of ice. There's water vapor, but there appears to be no liquid water. There are signs on Mars that there once was liquid water, and then it may yet be discovered that there are some places where liquid water does occur. But the planet itself, by and large, is desert. There is no liquid water available. Where there's no liquid water available, life cannot occur because all of the metabolic processes must take place in solution. So we need to spend a little bit of time talking about water and its properties because there's some very specific properties associated with water. It's the principal biological medium on Earth. Cells, in fact, are usually surrounded by water. Even our cells, we have a liquid a water-based medium which bathes all of our cells essentially. And if we go into the cytoplasm, we find 75–90% of cytoplasm is in fact water. The cytoplasm is a very concentrated solution of often very large molecules. The existence of liquid water and its abundance on our planet is one of the reasons why our planet is so special. Venus too hot, Mars too cold. We sometimes refer to as the Goldilocks planet because our conditions are exactly right. Water can occur in all its phases. It can occur as liquid, solid, and gas on our planet. And that is essential because that is how water is cycled around the planet. So there's something special about our planet. Water is abundant and life has arisen in water and is dependent on water. So water is a very strange substance. It has some really peculiar properties, and those properties are imparted to water because it is a polar molecule. And we'll see now that the two sides of the molecule have different polarities. They have different relative charge to them. And the reason for this is that oxygen, which is in the center of the molecule, water is H2O. The oxygen in the center is extremely electron negative. It is very, very attractive for electrons. It pulls electrons towards it. Now remember that water is a covalent substance, covalently bonded substance. The electrons are circulating, all of the outer electrons are circulating in a cloud around all of three of the participating atoms. But oxygen is strongly electron negative, and the electrons spend more time around the oxygen than they do around the hydrogens. They are pulled towards the oxygen. And as a result, the hydrogens, which have the electrons for less time, have a slightly positive charge. It's here described as delta plus. The delta charge is a slight difference in charge, and it's slightly positive for the hydrogens. Correspondingly, because the electrons which are negatively charged spend more time around the oxygen, the oxygen is negatively charged. And this is the polarity of the molecule. It is slightly positive here, and it's slightly negative there. And for that reason, the different different water molecules can begin to orient themselves. In water vapor, they don't do it. It's hot enough that in water vapor, the water molecules are independent of one another. In liquid water, the molecules begin to associate with one another because this delta negative here, this negative side of the molecule around the oxygen, is attracted to the positive hydrogens. There's a charge attraction here. So the molecules begin to turn and they begin to link together with a very weak bond called the hydrogen bond. Very, very important in biology. You need to understand that the hydrogen bond can arise in all sorts of molecules where we have a strongly electron negative atom that's pulling electrons towards it and inducing a polar charge on the molecule. In water, this is really important because, for example, with these weak attractions, they're weak, but there's a gazillion of them. So, for example, if we cool water vapor down enough that the molecules can begin to form these associations, we form liquid water. The number of bonds are high, but they're not terribly strong. And that imparts liquidity to the water, but it also gives water, because the molecules are attracted to one another like this, it gives water viscosity. So, for example, if you look at the surface of water, you can float things on the surface of water because all the water molecules are linked like that. And there is a certain strength to those linkages. If I cool water down tremendously, these bonds become very, very strong. The molecules move close to one another, and they start to form a crystalline structure, which we call ice. So all three of those are possible on our planet. Now, this polarity here has other very important properties. And one of them is, here's a depiction of water ice. The hydrogen bonds here are strong and stronger and much more stable. Liquid water, the bonds are somewhat weaker. The molecules of water have higher kinetic energy. They can break apart from one another more easily, and that imparts the liquidity to the water. These properties, by the way, are important biologically. We won't discuss them in much detail, but they are very important biologically as well. Okay, so the real major importance of water in biology is that it is a very, very good solvent. It's almost what we would refer to as a universal solvent, a substance that could dissolve anything. Not quite, but water can dissolve many, 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 many substances. Just by definition, a solution is liquid that has a homogeneous distribution mixture of substances in it. And the solvent is what does the dissolving. What we dissolve into the solvent is a solute. And an aqueous solution by definition is a solution based on water. So it's the reason that water is such a very, very good solvent is because of its polarity. And its polarity allows it to especially be a very good solvent of ionic substances. Because remember that ionic substances are those which have donated or accepted an electron from another atom. They have a charge. They are ions. Ionic substances dissolve as ions into water. And the reason water is able to dissolve them so well is because it surrounds the ions with a hydration shell with very weak hydrogen bonds which surround the ion. Here, you can see here, if we were thinking about table salt, NaCl, the Na, the sodium ion, is positively charged. It is a cation. And in water, this is what happens. The oxygens face towards the positive. They are attracted to the positive. You can picture, this is just in 2 dimensions. You can picture it's actually, they would be surrounded by a sphere of water molecules like that. The water molecules, in fact, can sort of pull them off the crystalline solid and dissolve them very easily. Chlorine is negatively charged. It's an anion. And so all of the hydrogens point towards, are attracted to it, and again, surround it to form a sphere around it, a hydration shell around the ion. And they isolate the ions from one another, keep them individual, and keep them in solution. That is why water is such a good solvent for ionic substances or anything which is charged. There is another property of water which we have to take account of, and that is a water's ability to actually form ions of its own. The water molecule can, in fact, split apart. This is overall not in the sum total of the number of water molecules we have. This is probably not all that common, but it does something very, very important. First of all, if a water molecule splits in 2, we'll end up with a positively charged hydronium ion, HHH. So we use 2 molecules of water in this dissociation. 1, 2, 3 hydrogens there, 1 oxygen there, and here an OH, a hydroxide ion. So in general, if I contribute positive charge to a solution, I will make it more acid. If I, in any way, I add positive charges to a solution, I'm going to make it more acid. If I add negative charges in the form of hydroxide ion, I'm going to make it more alkaline. So when water dissociates like this, there are as many hydronium ions as there are hydroxide ions. Pure water is neither acid nor alkali. It is neutral. But if I begin to add OH groups, say I added sodium hydroxide, NaOH, to my solution, I'd vastly increase the number of OH ions. I start to turn that solution basic. I start to make it alkali. Likewise, if I increase the number of hydronium ions, if I increase the positivity of the solution, say I add HCl, hydrogen chloride, hydrochloric acid, HCl. If I add HCl, I'm adding hydrogen ions to the solution. I begin to make that solution acid. And we will see that these changes, even slight changes in the number of hydroxide or the number of hydrogen ions in solution, will radically change the properties of the solution and may have big effects biologically. So this is something we need to watch really carefully. When we talk about solutions like this, we rate them on a scale. It's called the pH scale. And what you're actually doing in the pH scale is you're measuring the relative proportion of hydrogen ions, the pH. Neutral pH is 7. There are as many hydrogen as hydroxyl in the solution. If I add lots of OHs, I'm going to swing the pH. Now, acid is below 7. So 7 is neutral. If I add hydrogen ions to it and I increase the acidity, I drop it below 7 and go as low as 1, pH of 1. If I add hydroxyl ions, OH, to the solution, I will raise it, and I'll raise it above 7. So I can go up 11, 12. and that is strongly alkali. These changes in pH are biologically very, very important because they affect the structure of proteins, especially. A lot of proteins require a specific pH at which they work best. If you change the pH, they don't work well. So you need to get used to the terms. Acids, easy to understand. That's like a familiar term. That is any substance which effectively increases the hydrogen ion H plus concentration of the solution. It, in fact, increases the hydronium ion because the hydrogen goes to the hydronium ion, but don't worry about, just remember, if I add H plus, if I add a hydrogen ion to water, I decrease the pH, I increase the acidity. I know it's difficult to get that clear, but if you increase the acidity, you're decreasing the pH. If you increase the alkalinity, you're increasing the pH. I also point out, get used to thinking about this now. This ion, the H plus ion, is a proton because the hydrogen atom only consists of a proton and an electron. If I strip that electron off and I'm left with H plus, I'm just left with a proton. That's all it makes up a hydrogen atom, a proton and an electron. Take away the electron, you've got a hydrogen ion, H plus, same, it's a proton. We're going to hear a lot about protons as we start to talk about metabolism. So this is the so-called pH scale. And for example, here is the acidic side, and this would be less than 7, anywhere between 6.9 and all the way down to 0. This is the acidic, and the lowest that we ever see in biology is around about 1 or 2. And in fact, we have one area of our own bodies where the pH is extremely low. Can you think where it is? Of course, it's the stomach, because our stomach produces hydrochloric acid for digestion to aid in the digestive process. So the stomach is very low pH, around about 2. Most biological systems are around about neutral. Inside the cell, it's around about neutral. And these strongly alkali solutions are very rare in biology. But if I add a lot of hydroxyl ions, OH ions, to a solution when OH is very much greater than the number of hydrogen ions, that solution will be strongly alkali. Believe it or not, even in very, very acidic and very, very alkaline environments in nature, life can still occur. There are organisms which are adapted to living in very, very, very acid conditions and very basic conditions as well. Life can certainly occur there, but it's not common. Most biological systems function around neutrality. Just to give you an idea, gastric juice, around about 2. Sulfuric acid that you would have in a battery, that's about 1. You'll see here all bloods, that's all biological liquids around about neutral. Urine, saliva are slightly acid, not markedly. Seawater is slightly alkali. It actually says here 8, but it's actually around 7.4 to 7.8, around about there. And the alkalis do not commonly occur in living systems. High alkali solutions do not usually occur in living systems, but here you can see some of the things that you might have in your house, what their pH is, and you'll see all the alkalis are very high pH, 10, 11, 12, 13, around about there. The oven cleaner is actually sodium hydroxide. and that has a very, very high pH. It's extremely alkali. So this implies something. It implies that biological systems would not function well if their pH was very fragile. Now, you can, if I come here, and I'll show you, if I can very easily drastically affect the pH by adding very small amount of acid, for example. Just a small amount of acid contains a huge number, like a strong acid, contains a huge amount of protons, and I can drastically drop the pH just with a very small amount. Similarly, I could drastically raise it with just a small amount of a strong alkali. So biological systems require a very narrow pH range. And that is largely because the proteins require a certain pH to work optimally. It's not good for a biological system to veer from one pH to the other and drastically over a short period of time. Instead, the pH needs to be maintained within narrow boundaries. And as a result, biological systems very often depend upon a chemical system which actually prevents these radical changes in pH. Should something happen where suddenly a whole lot of hydrogen ions are added to cytoplasm or whatever, there are chemical systems which kind of soak up those positive protons and prevent the solution pH from dropping. Similarly with alkali, suddenly increase the number of hydroxyl ions. There are chemical systems within the living cell which attract those hydroxyl ions and bind them so that they don't contribute to a pH change. Substances which do that inside cells are called buffers. And buffer systems are tremendously important in biological systems because they keep the pH within a narrow range. There are all sorts of processes taking place in metabolism which could contribute, could cause especially things to become more acid than they should be. But buffering systems reduce the acidity and bring the pH back to an optimal level. And just remember, if you want to read up how buffering systems work, it details it in your book. Okay, so that's all I'm going to say about water. Now we need to move on and start talking about the processes of life itself, and especially about biomolecules. Remembering that this all takes place in solution, of course, but we need to address the first most important atom of all for living things, which is carbon. And carbon is this remarkable element which forms the basis of all biomolecules and forms the basis actually of all life on our planet. And as a result, we need to just try to understand why it is, what is this about carbon that actually makes it so important. And what it is that carbon is incredibly versatile. It is able to form many covalent bonds in different ways and allow different biological structures to be created. When we look at living things, most of our biomolecules have a skeleton of carbon. Often these molecules are extremely large. Carbon forms covalent bonds which are very, very strong, and that allows molecules to become very large, very complex, and yet to be very mechanically very stable. Everything we're going to talk about, we proteins, DNA, carbohydrates, lipids, these are all composed largely of carbon in one form or another. And so have a look here, and this is the carbon atom. This is its inner electron shell. That does not participate in any of the processes we talk about. But in its outer electron shell, it has 4 electrons. Remember that it wants, quantum physics says it should have 8. So it is driven to form 4 covalent bonds. It easily forms 4 covalent bonds with other molecules, and they are disposed in space in this kind of tetrahedron like this. And each of these would be available for a covalent bond. And so we say that it has a valence of 4. Its atomic number is 6, so it has 2 electrons in the inner shell, 4 in the outer shell. And this is the way that those electrons are disposed in space in their electron clouds. The electron clouds form that tetrahedron. And that is going to form the basis of the physical structure of molecules, of compounds which the carbon atom participates in. The study of compounds manufactured that use carbon as their basis in their manufacture, that is called organic chemistry. So an organic compound is any compound produced by living things which has carbon as its basis. The very simplest of all of the organic compounds is actually methane, and methane is CH4. So here's the C, and if you can imagine 4 hydrogens attached at each of these, 4 hydrogens, that's 4 more electrons, 8 in total, and that's CH4, and that is methane, the simplest of the organic compounds. And it is a gas, a flammable gas. And we'll see that basically everything becomes an elaboration of that, the simplest of all of the structures. Now, most organic compounds contain hydrogen atoms in conjunction with the carbon. And then there are some other elements, not a whole lot, but there are a few other elements which we frequently see associated, especially in biomolecules. That electron configuration is key to the structuring of biomolecules. And we're going to see that the actual physical structure in space is dependent on the way in which the carbon links up with other things. So here are the other elements that carbon can join with, that we will see it joined with, frequently with hydrogen, very often with oxygen, sometimes with nitrogen, especially in things like the proteins. But very importantly, carbon can bond to itself. It can join one carbon molecule to another. It can join, in biological systems, we will see it joined with either 1 or with 2 bonds, usually. And that makes a big difference to the molecule as to whether it's 1 or 2. But it's very important to realize that carbon can join to itself, because this is the way in which we're going to link carbon molecules together, and we're going to start forming a skeleton of our biomolecule. And then adding either oxygen, hydrogen, or nitrogen, that is going to change the properties of the molecule that we are talking about. So here's methane, the simplest. Okay. As we described before, CH4, and all the bonds here, each has a hydrogen attached. If I take that two methanes, I can actually join them together, because I can take out one of the hydrogens and use that to bond a carbon to another carbon. And that is this molecule here. It's called ethane, and also a gas, also a flammable gas. And here it's CC, so our first instance, of starting to form a carbon skeleton, CC, and all the other available bonds have hydrogens associated with it. Ethylene is somewhat related to ethane, but it has a double bond between the carbons. So now what we want to do is we want to count out how many bonds. So we've got 2 to hydrogens, 1, 2, 3, 4 to this carbon. Again, 1, 2 to the hydrogens, 1, 2 to the other carbon. You know, a double bond here. And here, this now is C2H4. So this gives you an idea. We can start to join carbons together, and we can join them in different ways. The way in which we join them is important. Single bonds are different to double bonds, and it has an impact that I'll describe in a minute. So these are the ways in which we can start to build carbon skeletons by joining the carbons together. And there are 4 principal impacts. There are 4 principal things that we will notice. First of all, we can change the length. We can add more carbons in the chain. And in biological systems, we often have enormous molecules with thousands, hundreds, if not thousands, of carbons joined together is possible. These bonds, remember, are very strong. So this is a strong backbone to these molecules. So we can change the length, the number of carbons that we join. If we put double bonds in anywhere, then something happens. It actually alters the physical structure of the molecule in space. It actually introduces a kink in the molecule. It introduces a bend, and it changes the functioning of the molecule. So have a look here. These 2 compounds here actually have the same number of carbons, the same number of hydrogens. They look very similar, but they have a double bond in different positions. This one has a double bond here. It's called 1-butene. It has a bond here at the first carbon. This is 2-butene. It has a double bond at the second carbon. And those would introduce a bend in the molecule, in the skeleton, and they change the properties. Remember, structure is strongly related to function. Change the structure, you're going to change the function. So if the cell is making a biological molecule, it will make a big difference where during synthesis a double bond was introduced. Thirdly, we don't have to have straight chains. We can have branched chains. This compound here, butane, has the same number of carbons and hydrogens as this compound here, which is methylpropane. The difference is that there is a branch. At the second carbon, there is a branch. And so this structure is quite different to that. The properties of this, even though they have the same number of carbons, same number of hydrogens, function is quite different. Fourthly, especially in solution, chains of carbons can form rings. And these become very important in carbohydrates and in nucleic acids. Just bear that in mind. That we very often see rings formed by the carbons looping back on themselves in this way, so that a single carbon here is forming bonds to its other carbons and pulling up a linear molecule into a ring structure. Remember, we're going to see these, especially in carbohydrates, and they become very important in the nucleic acids. There are other ring structures that apart from this, that we will see. Length, double bond position, branching, and presence of rings. These are our principal building blocks of shape for molecules made of carbon. Okay, so there are some chemical groups which we need to understand, which can be attached to these carbon skeletons, which impart very particular chemical properties. And I'll just tell you quickly that if I take a carbon skeleton and I'd all of the bonds are all of the available bonds are given to hydrogens. So I only have single bonds between the carbons, and then every other bond has a hydrogen. That we call a hydrocarbon. Hydrogen and carbon together, hydrocarbon. But those hydrogens can actually be replaced. They can be replaced by other elements, but they can also be replaced by functional groups. They can be replaced by chemical groups which have a specific character, a specific chemical character. These are hugely important in biological molecules, and it's what gives all of the character and function to biological molecules, is what functional groups are included. And there are 7 of them here: hydroxyl groups, carbonyl groups, carboxyl groups, amino groups, sulfhydryl groups, phosphate, and methyl. And I want you please to make every effort to learn these. Okay, we're not going to do them in a huge amount of detail, but I do want you to learn to recognize them, and then also have the principal character of the group. First of all, the hydroxyl group you already know. You can already guess what the function of the hydroxyl group is. Here it is here. This is OH, and OH, it very often imparts a slightly basic nature. So that's OH group is the hydroxyl group, and it occurs widely. We often see it. Typically, we see it in sugars, but also in things like alcohols. Here is an alcohol with its OH group there, and it imparts, because it's negatively charged, it imparts a polar nature to the molecule. So anything with the OH, you can expect it to be able to participate in hydrogen bonding quite easily. Here's a carbonyl group. We're not going to worry about it over much. Just be able to recognize it. It's carbon double bonded to an oxygen. It's a carbonyl group. If we were doing biochemistry, we'd spend a lot of time talking about it, because it becomes very important in sugars and things like that. Here's one we do want to keep an eye on, because we'll see this quite often. This is a carboxyl group, and the carboxyl group is carbon double bonded to an oxygen with an OH on it. It's OH, and this imparts an acid character. So this we'll very often see in compounds like organic acids, carboxylic acids, organic acids, things like that. As soon as you see the carboxyl group, you know that is imparting acidity either to the whole compound or to that region of the molecule where it occurs. Okay, the OH was alkali. Carboxyl groups impart acidic. Okay. This is an important group. This is an amino group, and this is a nitrogen. This would be attached to carbon somewhere in the molecule, but here is the amino group is NHH, so it's NH2 there. This actually acts as a base, and the reason it acts as a base is because it pulls an additional proton towards it from the water. So it removes a proton. If I'm removing a proton, if I'm binding up a proton, I'm actually raising the pH of the solution, because I'm removing protons. Remember that if we add protons to a solution, we increase the acidity. If I remove protons, I'm going to increase the alkalinity, the basic nature of the solution. So this is actually acting as a base, and a very important one in biological systems. This group here is called the sulfhydryl group. Just remember it because it occurs in a particular amino acid, and it becomes very, very important in the structuring of proteins. The sulfhydryl group, SH, sulfur and hydrogen. But if I bring another one here, I can actually bond them together with a covalent bond. Two sulfhydryl groups will covalently bond and form a very strong bridge. And in proteins, that's used to structure the protein. Phosphate groups, we're going to see a lot about these, both when we talk about metabolism and when we talk about nucleic acids. They occur in nucleotides. That's their most prominent role in the cell, but in all sorts of forms, not just in nucleic acids, and they're used in all sorts of different ways. So these are phosphate groups, and the one place that you really need to remember that phosphate groups occur is in a compound called adenosine triphosphate. Adenosine triphosphate is the most important energy source, immediate energy source for biological systems. That is, we often refer to it as biologically useful energy. And the energy is actually lodged in the bond which the phosphate group makes. So the adenosine triphosphate, there are 3 phosphate groups. The phosphate groups are all joined together with covalent bonds. And the biologically useful energy is mostly located in that first bond and in the second bond, but especially in the first. And if we break that first phosphate bond, we can extract the energy and use it biologically. So these phosphate groups are very often associated with a transfer of energy. We'll see a couple of other roles as well. They're often associated with the transfer of energy. This is a methyl group, and these methylated, when you add a methyl group, you refer to it as methylation. And methylation is often a very important control mechanism in biological systems. It's often used as a switch, to switch systems on and off, where you can methylate them. For example, it's used in DNA. Methylation has specific impacts on gene regulation. We also often see methyl groups associated with things like hormones and things like that. Okay, so for the moment, that's it for today. And then Wednesday we'll carry on. We'll be talking specifically about kinds of biomolecules, okay? This may be a little bit shorter than our normal lengthy class. Use any extra time to repeat this Zoom class and to go over especially those functional groups. Use your textbook to read up a bit more about the functional groups. And apart from that, I will see you on Wednesday.